Hello everyone, and welcome back to our next lecture in regards to our unit on cell division. This video's topic is going to be the process of mitosis. So we're going to start with this next slide right here. And this should look familiar to you from the last video we did regarding the cell cycle, which we defined as the process, pretty much the life of a cell as well as its division eventually at some point. Right now for mitosis, our topic is going to be on those types of cells that we refer to as somatic. A somatic cell is a body cell. That means pretty much anything in your body that's not either a sperm or an egg cell. And if it's a human somatic cell, you should know by this point that it has 46 total chromosomes in it. So our focus right now is on a cell that's just been born, if it's a somatic cell. It enters G1, so it continues to grow and get bigger, but at some point it may pass what we call the restriction point, or point of no return, where it's gotten too big, so its rate of diffusion is going to start to suffer. So if it can, it'll enter the S phase, where it'll copy all of its DNA. From there, it'll move on to G2, which is it proofreads and double checks all of its DNA, make sure that it copies every single organelle that the brand new baby cells are going to need, as well as it'll make some additional food and proteins for the process of cell division up here, which will be mitosis. Mitosis, you can see, is going to be broken down into 4 major stages. There is a second point to division called cytokinesis. That's not shown here, but we'll get to that shortly. All right, so let's begin. Mitosis, by classical definition, refers to the division of a cell's nucleus. So that does not mean that the cell will literally split into 2 individual cells. It's really all about taking a nucleus, which you can see here, copying all of the DNA, and then you go through several steps. You can start to see the chromosomes actually forming now, until eventually that original nuclei divides into 2. In this picture here, you can see 1, 2 nuclei as opposed to the original one up here. So technically by this point, mitosis would be finished, and a second part of division called cytokinesis would actually separate these 2 cells here into individual ones. And what you're looking up here right now is actually colorized muscle cells that they were taken with an electron microscope. So let's begin the process of mitosis. We're going to start with the first part of mitosis called prophase. Mitosis, I always try to remember it as PMAT. So prophase, metaphase, anaphase, telophase. In short, PMAT. So starting with mitosis, we have prophase. There's going to be several key things that you should be writing down on the notes that I posted for you to remember what happens here during prophase. The first is all of the DNA that, if you remember, existed as spaghetti or chromatin, starts to coil up and condense into these thick rods that we call chromosomes. And by the time we can actually see the chromosomes, they're going to exist as these big, thick X shapes that we can see here where I'm circling. Remember, before division occurs, all of this DNA had to duplicate. So when you see an X down here, what you're actually looking at are the same chromosome copied 2 times. So each half of the X is an identical copy of the original chromosome, and they're currently attached in the middle by what we call the centromere. So if I come back up here to this slightly larger picture, so this half of the X is 1 chromosome. The other half is an identical copy also of that same chromosome attached in the middle by the centromere. So let's not forget here that as long as 2 halves of this X are connected in the middle, we're going to call that 1 whole chromosome. Then we have 1, 2 chromatids. A chromatid is a copy of a chromosome, so you can see each half of the X is a chromatid. And then the whole X together we refer to as a sister chromatid pair. All right, we'll touch more on this as we proceed. Second key thing that happens is all the chromosomes, as I just mentioned, will be paired in that sister chromatid pair form, connected by the centromere. And our third key thing that happens here in prophase is the nuclear envelope or membrane disappears. It'll just dissolve away. So over here you can see that the dotted line is the nucleus starting to disappear. You can already see the chromosomes right here at this point that have fully formed. All right, over here in this current animation, here is the nucleus starting to disappear, and all the chromatin condensing to form chromosomes, and eventually you can see those X shapes right here that start to form as the process proceeds. So again, all the DNA coils up to form chromosomes. We'll see them in these X shapes. This will be one chromosome, one sister chromatid pair, or 1-2 chromatids. And our picture over here, as a control, we're starting with a total of 1, 2, 3, 4 chromosomes. We move on to the second phase of mitosis, which is metaphase. Metaphase has several characteristics that we need to remember. First is all the chromosomes, or sister chromatid pairs, those X's, are going to line up along the equator. That means the middle of the cell. That could either be in any picture from top to bottom, or it could be from left to right. Either one is fine. So you can see that in this animation over here, that all the chromosomes, 4 of them, because there's 4 X's, are slowly lining up here along the middle of the cell, or the equator. Next thing that's going to happen is we're going to have little proteins called microtubules. The official expression is spindle fibers, but spindle fibers or microtubules are the same thing. Those are these little lines you see down here in the cell. They're going to connect to a part of the centromere, that center part of the chromosome X here, called the kinetochore. So if this whole middle region here where my cursor is a centromere, the actual part where the spindle fibers or microtubules attach to is called the kinetochore. All right, and these spindle fibers are being, like little spider webs, are being shot out of these little structures here. These are what we call the centrioles that we learned about briefly way back in our organelle unit this year. So as the chromosomes line up along the middle, these spindle fibers that are being emitted by the centrioles will attach to the part of the centromere called the kinetochore. Third stage of mitosis is called anaphase. During this part of mitosis, all sister chromatid pairs are going to split. So you can see down here, as it continually recycles back the animation, the X's are being pulled apart by the centrioles. So it's kind of like a tug of war. The spindle fibers are ripping the sister chromatid pairs apart. So they're separating these X's. One half goes to the left, one half goes to the right. So you're going to see that pretty much you're going to get just one version of each chromosome moving to an opposite end of the cell. Now, one thing to take note here, during anaphase, once the sister chromatid pairs split, my chromosome number temporarily doubles. So if I count my centromeres, now I have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes, not 4 like I started with. Because the goal is to get one cell over here that'll have just 4 single chromosomes, and one over here that'll have 4, just like the original parent cell. And if we now move on to the last stage of mitosis, which is telophase, several things we need to know about telophase is, first, the nucleus is going to start to reappear. So the dotted line represents the nucleus or nuclear membrane reforming. Over here in this animation, you can see that start to happen right here, which are these little yellow dots representing the new nuclei or nucleus reforming. You can see you're going to have 2 of them. All of the chromosomes, the big thick rods that we see that split during anaphase, they're going to uncoil back into chromatin. So see how they're getting elongating and start to see more of a solid dot here? You don't really see chromosomes anymore once you reach the end of telophase. So right now you can see pretty much you have 1, 2 cells that have formed. We have 2 brand new nuclei. So that means technically mitosis is officially complete. You have taken the original nucleus that had 4 chromosomes, you duplicated those chromosomes, and now you're going to have, again temporarily, 1, 2 nuclei that only have 4 single chromosomes, just like the original cell that we started out with in this example that had 4 chromosomes. If this was one of our cells, a human cell, you'd now have 2 cells that have 46 total single chromosomes. So to get these two cells now to finish division, we finished mitosis, dividing the nucleus, but to finish division, now we got to actually separate them in two. Almost got to like kind of cut them right down the middle. So the last thing that needs to happen is something called cytokinesis. So this will be the second part of division. Mitosis was the first. What we're going to do now is we're going to separate the two brand new cells that we've made, the daughter cells is what we call them, by splitting the cytoplasm in half. Pretty much we're going to split the cell membrane and divide the cytoplasm in half. This happens a little differently in animals versus plant cells. If we focus on animal cells first, since that's what you and I are, this will be down here, the end of telophase. You can see your two new nuclei have formed. Now you're going to start to separate the cytoplasm of the two cells here. All right, the cell membrane starts to pinch inwards and eventually divides the cytoplasm in half. And you get two brand new individuals, see how there's space, daughter cells like you see over here. And once this restarts momentarily, you can kind of just look at that again on how that happens. In plant cells, they do divide in half, but they don't fully separate into two individual cells. What plant cells do is they start to grow a new cell wall. Alright, so they're not pinching the cell membrane in half. They just grow a new cell wall, sometimes called a cell plate, and that'll officially separate the two daughter cells into individuals. However, they're still connected by a thick cell wall. Now that's important because plants, think of a big, huge, gigantic tree that might be out in your backyard. They need strength to support themselves so they don't just fall down or fall apart. So when they do become separate cells, they're still connected to each other like glue by a big, thick cell wall for strength. We don't really need that as much. We have our bones to support our overall body. So in this picture here, what you can see is you have just a general idea again of prophase. Your chromatin condenses in the chromosomes. I can see 4 chromosomes or 4 sister chromatid pairs. Eventually you get to metaphase where they line up down the middle of the cell. The spindle fibers will have attached to the kinetochores on the centromere of each chromosome. Again, you have 4 chromosomes or 4 sister chromatid pairs, or 8 chromatids total right now in this cell. Anaphase, it's the sister chromatid pairs, the X's that split. Technically now I have 8 chromosomes, 1, 2, 3, 4, 5, 6, 7, 8. And eventually telophase occurs, the nuclei reforms. You can see there's only 4 chromosomes now in each individual cell, just like the original parent cell had. Alright, they just haven't been duplicated yet over here. And the final stage is cytokinesis will split those cells in half so you get 2 brand new individual ones. This is how it would actually look in one of your cells in your body right now. Solid nucleus during interphase. Get the prophase, you can actually see the chromosomes. Skip ahead to metaphase, lined up in the middle. Anaphase, the sister chromatid pairs split. Telophase, you can see how cytokinesis is also starting to happen here. But for telophase, see how you don't see threads anymore? All the chromosomes have uncoiled back in the chromatin and the nucleus is starting to reform at this point.